Good evening, everybody. So, Waka, should I start right now?
Yeah, hi Maksud. Yes, Maksud, you can start with. Yeah, thanks.
Yeah. So, I have always been work with the bacterial genome. So, this is, I think, uh, I had very few, little experience with the human genome data, but I try to gather the information in the last several hours on this databases and the disease-centric databases. Hope this will be useful for you. And it's very difficult to go through the like hands-on within the one lecture, but I'll try to cover as much information as possible. So at the beginning, we'll cover the population genomic databases, HapMap 1 KB databases, gnomAD and ExAC. Then a couple of databases, disease-centric databases, OMIM and Cosmic, which are very well known to all of you, I guess. So international HapMap projects, actually, before that, the HGMP project started. And the first human genome was sequenced around it was announced in 2001. And at that time, the human genome mapping project started to annotate, complete the annotations of the genome in a consortium, and the USA, UK, and all the scientists from the many other countries actually worked together to annotate the genomes. Then the HapMap project started, and the goal was to determine the genomic variations among the genomes from the different regions, and is there any common sequence variations among the genome taken from the and to make this information available in the public domain. And the different samples are taken from the different ancestry, including the Africa, Asia, and the Europe. This is the paper was published in the 2003 in the Nature. And they anticipated the discovery the sequence variances affect the common diseases and should facilitate the development of the diagnostic tools, common targets for intervention, and the drug discovery. And we can see that now we're moving to precision medicine. So there are few important publications came out from these projects, the HapMap projects. 3 major publications came in the Nature. The one in 2005, 2007, and 2010. And phase 1, phase 2, and the phase 3. And then they're followed by the 1,000 genome projects. Around the 2010 actually, so the next generation sequencing or the high throughput sequencing started to bloom, and that resulted in the planning of carrying out the large scale genome sequencing projects that followed the idea of like the started the 1,000 human genome projects, and that was kicked off around 2010, but it increased gradually. You can see in the 2005, in 2012, one paper came out with the 1,092 genomes. Then in 2015, in 2015 came out to the 2,504 genomes, and there are larger genome projects going on all around the world. And one of the UK biobank, they have represented they're planning to sequence 1 million human genomes at the moment. So that's known as Genomics England. I didn't include this here. Okay, as I said, the goal was to develop a haplotype map of the human genome and to find the common patterns of variations among the human genome from the different ancestries. And the idea was that because and to find out the variations that might affect the health, disease, and the response to drugs and the environmental factors. And it officially started with a meeting held from October 27 to 29, 2002, and the goal was achieved within the 3 years. We can find that it was in collaboration with Japan, United Kingdom, China, Nigeria, and the United States. And the list of participating organisms can be found in this link. And obviously, this is very important. We know that a lot of paper came out that the multi-omics would say not the genomic, only the genomic variations, but they have the aim of the HapMap project was to find the relationship, whether any relationship with the variations of the different diseases like the diabetes, cancers, heart diseases, stroke, depression, asthma. And although we know that the two human beings actually share 99.9% of the DNA, but the remaining 0.1% is huge actually. So that might be responsible for the different diseases or the different outcome, phenotypic outcome. And as we know, the discovering the DNA variants can contribute to disease risk factors. For example, the different risk, the cancers, diabetes, or different other diseases. So I know you know about the SNEs, but there sometimes the confusion happens. What is haplotype actually? Haplotype actually, so we know that the 10 million SNP exists in the human population, for which only the rare SNP is only 1 in 1,000, so 1%. Yeah. So alleles of SNPs are close together, tend to be inherited together. In the next, I'll show you the example what it means actually. The SNP. So the SNPs actually, for example, since they're closely located, they tend to inherit together actually. And this association, the changes, associated changes is called the haplotypes. So most chromosomes have only few common haplotypes, and which account for most of the variation for person to person. Let's see how it works. For example, as he said that the SNPs actually, you can see there is a SNPs A to G and the T to G, for example. And this basically the changes actually, the nearby SNPs actually change quite frequently, and this is called the haplotype. For example, AT is an haplotype, and GZ is another haplotype, AG is another haplotype. So this changes, the SNPs, they're like located closely actually, they tend to be changed to inherited together more frequently than the others, and this is called the haplotype. And obviously, the lot of huge studies going on to find the genes that affect the diseases such as the diabetes. And there is this is basically taken from the ideas taken on the 2012, and they had a plan to find the disease specific markers, and they could also find the chromosome regions. Two groups different the haplotype frequencies might contain gene effective. So haplotype frequency might have an effect on the genes, and that contribute to the regulation of the different diseases. So what kind of populations are samples of the HapMap projects? So you see that the common haplotypes occur in the, so if you compare all the samples in the databases, you'll find that there are common haplotypes. There are some haplotypes very common, like common to 80 to 90%, but however, their frequencies differ in the population. For example, one haplotype might be found 80% time in the African population, and in another population that might be 70% time. So they differ among the different populations. And pilot studies have shown that there is a country geography specific frequency of the haplotype frequencies among the different population for different population in the different countries. So this basically necessitates the study in a very larger scale. So as you know, the most of the sequencing has been done from the developed countries. So we need to start sequencing to see the pattern, the haplotype pattern, and this is a specific marker for the these regions. So in the phase 1, the 269 people was taken from the different countries. For example, 30 from the Nigeria, to the Japanese 45, Chinese 45 from the Chinese, and the northern US 30 trials was taken, and they're given their consent to carry out the studies. Okay, and Wakas has explained very elaborately about the DB-SNP. So through the HapMap projects, so the 3 million SNPs were found across the human genome, and 2.6 million are common with the DB-SNP. So there are some like unique SNPs are found using the HapMap projects. And the genotyping is carried out in the 11 centers in Canada, China, Japan, and the UK. And the phase 2, another so number of samples increased around, and the 2.1 million SNPs were genotyped in the samples. And I'll show you the method they use actually. And what are the policies concerning? So the policies that it should be publicly available, and it does not include any genotype phenotype relation for the disease associated genotype phenotype association data sets. And participants in the project do not believe that the SNP genotype or haplotype data for which generator are appropriately patentable. But if somebody is working on the data and he finds that there's some interesting SNPs there that's patentable, they can patent it, but they cannot restrict the entry or the, because that SNP is always available in the data in the databases, so they cannot restrict the use of that SNPs, but they can patent that if they have found interesting data, so they want to patent, they can do that. And there are a lot of publicly available reference population databases, and some of them are shown here. For example, GME. So that contains the exomes from the individuals of Middle Eastern ancestry. So this is the only database where you find the genome data, exome databases from the Middle Eastern populations. And we're talking about the 1,000 genomes. And this is one of the first reference databases. And here you find the exomes as well as the low coverage genome sequence from the diverse ancestries. We'll come to the other databases later on. And this is one of the very first paper article published for the 1,000 genome projects. And you can see that underneath the low coverage whole genome of 170 individuals from taken. And in this study, they describe the location, allele frequency, local haplotype, and they identified the 15 million single SNPs identified, 1 million insertions and deletions, and 20,000 structural variants, most of which are previously undescribed. So 95% of the current lexical variants found in any individual are present. So 95% were common to all of them. Each person, and it's found that each person is found to carry up to 250 to 300 loss of function variants. 50 to 100 variants previously implicated. So another article came out in 2010, and in this one, so they, because in the beginning, the scientists were more concerned about the common variants in the human population. Then they said that there are a lot of rare variations among them, so they integrated the common and the rare genetic variations and tried to find the genotype phenotype correlations. So they generated 1.6 million common single nucleotide polymorphisms in 1,184 reference genomes from 11 global populations. And what they did actually, so here, okay. So as we said, that's only the common DNA variants are basically analyzed in the Human Genome Project, the phase 1. So in the second phase, actually, so they use allele frequency less than 5%, that is present less than 5% of the population. So, and they try to find the SNPs as well as the copy number polymorphism. So how many copies are different, polymorphic copies are found in the each population were also analyzed. And the study was validated using the polymerase chain reaction in the selected genomic regions. So the study is extended to 1,184 samples from the 11 population, as I said, and these are the areas from where the samples are taken, locations. The method is FMatic, that's a microarray method used, FMatic human pairs, and the Illumina human one single bit chips are used. So they're very specific genes actually, so, and they interrogated 1.8 million sites, and this bit chip interrogated 1.2, almost 1.2 million sites. And by filtering the low quality data sets, so it might be difficult to understand you at the moment what is the low quality filtering. So when you understand, when in the next phase, when you go to next generation sequencing data sets and you'll be analyzing, so you'll understand the low quality data sets are there. So if you like filter out that, the total number of variants are found after filtering around 909,622 from 1,326 samples. in the FI matrix, and almost 1 million sites from the 1,211 samples from the Illumina microarray technique. And between these two technologies, the concordance was 99.5%. The consensus between these two methods, the total number of SNPs were called 1.4 million, and one polymer SNPs are found in 1,184 individuals from 11 populations. So I'll not go into the details of that actually. So summary, what it means actually. So from the 1,000 genome projects, collection of samples from the 5 populations at the very beginning. And in the second phase, as you can see, they broadened the reference population because the population has been taken from the different sizes. That's a very good that's a very good database for the reference population to capture the geographical local variants. And that can use the robust reference panel. For example, if you're working on in the genome sequencing data projects, actually, so if you want to compare your data with any specific geographic location and find the unique mutation in your samples or the common mutations in your samples, you can compare your data with the data available in the 1,000 genome projects, because that's because there are a huge number of samples there, so that serves as a good reference database. So as we already mentioned about this, so pilot analysis. So first of all, they map the human genome variations common to all populations, then integrated map genetic variations found from the 1,092 samples, and the global reference map was generated using the 2,504 human genomes. So some more paper came out. So as I said, that a global reference for human genomes. So later on, 2,504 individuals are sequenced from the 26 populations. Earlier one is around 1,000 from the 11 population. So it's more robust. And they found in it, so there is a, so what he found the variant, and in this study, apart from the only the genomic SNPs in all over the genomes, in this study, they try to find the functional variations in the genomic locations more precisely, and they found that around the 200 sites with protein truncating variants, 10,000 to 12,000 sites with peptide sequence altering variances. These are, and African genomes are consistently high end of these ranges, had the higher variations in the African genomes. And that's obviously, so from that study, they came out that the origin of human being from the Africa, and observed 200,000 variants per genomes per sample, and the 24 to 30 variants per genome implicated in rare disease. So CleanVer is a database where you can find the association between a specific variation and the diseases. So per genome, actually per human being has the 24 to 30 rare variants that can be found in the CleanVer. And the Europeans have the higher rate of this genes available in the CleanVer. It could be possible that there are a number of genome sequenced more from the European ancestry. So that could be the reason. So you get even more variants, more of their variants available in the CleanVer clinical databases. So 0.7 million variants are rare. And we already discussed that some populations have a high number. I'll not go into details of the drugs. Yeah. Okay. So the 1.6 million had frequency of less than, so rare variants are only 1.6 millions, and maybe missing from the current data sets because there is a rigorous filtering methods. So let's have a look into how the 1,000 genome projects look like. Where can I find it? So International Genome Sample Resources here. So if you want, you can download your data from the FTP sites here. How to do it? So you can use the Aspera. It's a data. We can go to the data portal. Yeah. So you can filter by population here, what populations you want, and you can use whether you want the what kind of 10x genomics data sets, so packed by Oxford Nanopore data sets. I'm looking for the like packed by gives a longer read. So there should be some called the Illumina data sets. And so you can download. It's a huge data set. So once you have an idea about the next generation sequencing, you can work through this data sets. You should also be able to download the variant files and the genome mapping files from the 1,000 genome projects. We look in the gnomAD in more details. So you can play around with this data sets once you have an idea about the next generation sequencing data sets. So now comes the genome gnomAD. So that's because we have very interesting features in the exome. People are more interested in the exome analysis, and because they contain median rare and the potentially functional variants. So in the intronic regions, they don't have much effect on the functionality of the proteins translated. So as you can see, there are 500 rare mutations found in one. This is basically statistics about the usually found in the one genome, and 100 low frequency variants, 20 homozygous, and 100 rare variants found in the known disease genes, as we said, available in the clean burr, and around 50 reported disease causing mutations. So that's obviously needs to be experimentally validated, and 1 to de novo coding mutations means, so that's specific to that particular pertinent to a specific genome. So we'll see the gnomAD databases. As I said, I've always been working in the bacterial genomes. So I do not have much, but I'll try to explore as much as possible. So using the large reference databases, try to identify the rare variants in gnomAD. So you can see actually, so variations in East Asian populations, the high number of variants retaining after filtering, but after you filter that out, actually in the ExAC databases or the genome databases, there is a 5-fold reductions. So they use a lot of filtering options, like they took out the low quality reads, the homopolymeric side, the massive repeat regions, and after that filtering those low quality genomic sites, variation sites. So they count the 5-fold reduction in the candidate variants for further review and the exome analysis are found. So there is some publicly available reference population databases. I have shown that first. So one of the first reference databases, so exome sequence individuals of European and African ancestry. You can find the European and the African ancestry sequencing in these databases, common cohorts, and Discover HR. So you can see that. So this is a collaboration between the Regeneron Genetics and another health system brings together. And one of the good things about this site is that they have, by sequencing of more than 50,000 genome participants, they've identified more than 4 million rare single nucleotide variants and insertion and deletion events, and 176,000 predicted to result in loss of gene function. So that's the deleterious effects of the gene loss functions. And we can go to the Discover EHR. Just give me a second. So, okay. So for example, you can put the chromosomal positions of the RSA of the gene name and see how things work. So for example, you can say CYP4P1. So you need to accept this one, and you can see the variations and the compared to the reference genome. So they might be using version HG19 or HG38 human genome. So you can see the very references and the variation positions. This is the DB-SNP ID. So you can see that in these genes, what are the variations defined here. You can go to the details. Let's see this one. It's supposed to get the details of this one. So, say for example, so this is a I don't know, I can show you. So it's not showing anywhere. So it's just showing the positions where you have the variations and the frequency here. So this is a high frequency. You can go to the clinical databases and I can check how this affects the phenotype of and whether that has any pathogenic effect or not. So some other databases, for example, the OpenSearch, another one. So data from the human. So in this databases, the rare variations are documented, and you cannot download the datasets, but can upload your VCF once your datasets So as I said, that when you analyze, you'll be analyzing the next generation sequencing data. So variant file is called the variant calling format. So that's a BCF format. You can upload your variant calling file there, and you can do the annotations there, but you cannot download any data from there because there's a like the people who participated in the program, they didn't give the consent to share the data. The data will pay. So publicly available reference populations, and this is the first aggregated exome reference databases, and that represents the 5 ancestries. You can see here the South Asian, European, East Asian, Latino, and the African population. So if you're using the ExAC and genome data, so it's not advisable to use both the data sets because you have a lot of overlapping data sets. So it's better you either use the ExAC or the gnomAD, because gnomAD is a high, and now they're, so far I know they have been combined together. So it's better to use the gnomAD databases. So Bravo is a, here you will find only the whole genome data sets. You don't find the exome data sets here. And so restriction, so you cannot, it's the same way as the open search. So you cannot basically, you cannot download any complete data sets from here because of the constraint in the consent from the people participating in this consortium. So now we look at the gnomAD. So many PIs, principal investigators from more than 100 countries, more than 100 PIs, they participated in the study, and the number of people, individuals, 138,000 individuals, including the 123,000 exomes and the 15,496 whole genomes. Data are available here. And you know, the latest technology like the Illumina data processor, the same pipeline used. So for data analysis, they use the same tool so that the variation among the different sites are minimized. And they're called jointly, and most probably they use a pipeline, the GATK pipeline, that's developed by the Broad Institute in MIT. So you can download the variant files into data sets and can annotate, you can annotate your data sets with the allele frequencies, and individual level data not shared and phenotype data are not available, but there for some studies they're available, but not most of them. And no Mendelian disease, that means the gene phenotype of the gene and disease relationships are not available. And the new populations, because they had confounding factors, they basically separate out from the rest of the population when they do the PCR analysis. So that has been left out. And from the gnomAD, you can see the interesting thing that from the SNPs, so they did a principal component analysis, and you can see that the different populations clustered in the different areas. For example, here the orange, the Latinus clustered together, so and the East Asian clustered together, East Asian clustered together, Finnish population clustered together separately. So you have the different cluster of populations. Yeah, and the quality control they maintain is the lower quality samples, and the sex chromosome abnormalities and the first and second degree relatives were removed from the data sets. So because they might create the they might affect as they might act as a confounding factors there. And these are the populations. Here's the 1,000. The data was taken from the different sites. Almost all the sites database, 1,000 genomes. The Fin data sets. Go to the databases. Among them are the Sigma databases and Cancer Genome Atlas. This is another interesting data set. So there. So, and some of the phenotypes from the cohorts include gnomAD, for example, population between the population that the twin data sets, how they're related, are different. So, the genotypes of the type 2 diabetes, bipolar schizophrenia, migraines, TCG germline samples, and subsets planned for release, cancer cohort excluded. And yeah, so they might be released later. Diabetic cohort, the neuro side, because they have the different other data sets for it, like the cosmic ID. Okay. So let's have a look at how the gnomAD looks like. So, screen. Say, for example, you just give the name. All right. Okay. So you can see the sun transfer genome build. So that was mapped against the human genome 1819. That's in symbol IDs. So you know very well about this one because what is, so then there are how many synonymous mutations we found, missense mutations are found, expected, and how many observed in this gene. And so the green one is the sequencing coverage. As I said, so the next generation sequencing has the coverages. So and the blue ones shows the coverage in the exons. Yeah, exome sequence data sets. This is the genome variants actually. So you can look like the gray ones are the the yellow ones are the missense mutations, and you can see the green ones are the synonymous mutations. So if you click one of them, it takes you there, like the positions. What, for example, if you click here, it shows you here like we go down. So there are the mutations in these regions. So you can go through them. This is the number of alleles we found in the populations and how many mutations we found. So if you divide this one with the number of alleles, you get the allele frequency there. And say, for example, you can find, for example, the stop gain in these positions. If this mutations happens in the 722 position, glycine can be so termination codon. So that's pathogenic. If you click here, so this takes it the clean word genomic variation is related to human health. Review status that is reviewed by the expert panel. Yeah, protein change. So Q to 720 stop codons we find here, and how they link to the other databases. So you can go there and link to the DBSNP databases and submit, and the interpretation of the evidence is reviewed by the expert. So this database is very useful for the physicians. I think nowadays many physicians are looking into if the drug is not working for any cancer. So you can do the sequencing in specific locations, and you can go to these databases and look the implications of these mutations. For example, these mutations has an implication of cystic fibrosis. And this is the details about the submitter and what are the evidences available here. So you can click here and you can go. And I think there are some databases where you can see that what resistant, if they have an implication, they're resistant to the any drugs or not. So this is a very useful database, gnomAD. So anything else here? Yeah. And obviously you can go to the clean-but databases. And you can see the expression, the different tissues. So it's a resource developed by the, yeah. So they have, we have already said this once. can see it's, and there is some, sometimes you might find some flags when you're analyzing your data sets. So there might be some reasons where you have the low confidence of the, for example, they're saying that, okay, there's a loss of function in the proteins. Yeah, but they might be at that protein truncating region, so that might not be sequenced properly. And there might be some problems in the annotations, so the dubious transcript annotations. So, and sometimes you have the poorly, if you align the regions, poorly aligned regions, so that could happen where you have a lot of repeat regions. For example, you can see the lot of cysteine repeat regions. And once you call the variance in these sites, yeah, if you call the variance, you do have very low confidence. It's shown by the LCR. You see the low confidence regions. And sometimes if the homopolymerase, it could be because of the indulgence reasons and which for PCR artifacts. Yeah, but also it could be true. It could be true that you have the homopolymers here, but it's very difficult to sequence those areas. So the difference between the gnomAD and the ExAC. So 98% of the sample in the ExAC are in the gnomAD, as we have shown, because gnomAD basically is very comprehensive data sets. And so in the gnomAD, the data is highly rigorously processed in the ExAC. So it's better we can have the more data in the gnomAD. And this is a very old version. What I'm saying, actually, gnomAD is a bit matured right now compared to this information. And I think they have the final release right now. So, and the ExAC has been amalgamated with the gnomAD at this moment. So we can rely on the gnomAD apart from going to ExAC databases. I think most of you are familiar with the online Mendelian inheritance in man, so OMIM databases. Yeah. So as I said, I'm just going to the last several hours going through these documentations. I'm not very familiar with these databases. That's why I copied, put all the information here possible for you. So OMIM is linked to the external links, gene entry, phenotype data, genomic data, and also linked to the external data. And the dashed line basically, not all phenotypes are mapped to the genomic data sets. And so the output you get from the OMIM, actually it has certain interesting feature content. For example, it has a meme ID, and if the meme ID is followed by an asterisk sign, sorry, if the asterisk is followed by the meme ID, so this means it's a gene, and if it's a like hash sign, it's a phenotype ID, and the plus sign before an entry number indicates that the entry contains description of a gene of known sequence, known phenotype, and the percent sign means confirmed Mendelian phenotype or the phenotypic locus for which underlying molecular basis is not known. And there is no symbol actually, so that means it's been updated or deleted. Sorry, description of a phenotype for which the Mendelian basis, although suspected, is not confirmed, but it's suspected phenotype correlation. And the caret before an entry number means entry was removed from the databases or moved to another entry. So you can put, for example, potassium channel, whatever it is. So you can go through the gene map table and the clinical synopsis tables from here. They can see how many entries are there, and you can see the red button says that's a OMIM ID. Star means this is a gene, and you can go through the details of this one. So each entry, I'll just go through a little bit of things. So meme, you have a meme ID, as I said. So it has a prefix start sign, and you have the color triangles, so you can find gene and phenotype correlation. So if you click on the title, for example, it's a meme ID, and it will open the full view of your data sets. So you can go to the details of the gene you want to go through. So this is the HGNC approved gene symbol, this one, and the location in the cytogenic locations, and the coordination of within the, for example, the chromosome number 12 and the locations of this gene within the genome. You find, yeah, phenotype. You can find the phenotype. You can find the related text descriptions. Not going to the details, and yeah, so you can navigate through. So you can find the number, phenotype related, the dbSNP identification number, and go to the clean bar to find the relationship with any diseases or not, known diseases. So I'll just highlight on the cancer genome databases. So cancer is a very complicated data, so I'll not get into the details because I'm not a physician, but I'll discuss in the perspective of the databases available. So lots of databases, and you can basically come out with a better solution from this consensus from the different databases here. And one of the very known is a catalog for somatic mutations in cancers. So here you have like, one of the problems with the cancer is the somatic mutations. They're very much specific to the particular patient. So they're developed and curated by the Wellcome Sanger Institute. So more than 10 million curated. So here you have the different data linked to different information, cancer gene census, cosmic 3D protein structure, the effect of the mutation on the 3D structure. And you can go to the cancer browser. So if you click on the cancer, for example, if you type gene name, for example, if you type the ABL1 gene, yeah, it will take you here actually. And on the top, so it will show you the, so it will show you the rare, I don't remember the blue one, the red one, but it will show you the top 20 genes that are affected for that cancer. Okay, yeah, I got it. And how many populations have this, how many mutations are found in the total number of populations. You find the blue line, and you can find the mutational signatures here. Yeah, if you go to the ABL1, and you can find all the informations about this one, and you can, they're different, they're basically categorized in the different types. For example, this yellow means that this belongs to the tier 2. If you find in the red ones, so those are like the tier 1 signals for the responsibility like cancer. And this tier basically, the category is based on the whether they're the suppressor of the growth or they're promoting the growth of any particular gene cell. So this is how it looks like. So you can search for any genes. There are other databases, for example, the cBioportal. So this is a very useful one. And the good thing about this is that you can put your own data and you can visualize your data sets here. There's also, so you can see the lollipop graph. So that means that the higher mutation, the locations where the mutations rates are very higher, you can see those positions is in the cBioportal. Oncos KB, that's very useful for the physicians. They can, because they integrated therapeutic diagnostics and prognostic information along with the variant data sets. So they give the information to whether that's a FDA recognized or not, standard care from the different standard databases. So clinical interpretation of variants in cancer. So that's done by the McDonald Genome Institute in Washington University. And of the key collaboration between the somatic cancer and the CWC, a recent integration of protein paint. I don't know, I don't have much idea about, but I thought that it would be useful for the doctors to know about these databases. So this is almost look like the OMIM databases. The variant, interest ID with the which disease is related and what drugs can be used for this kind of mutation, specific mutations, and evidences linked to the external evidences, whether they have any evidences of these variations or not. So they also provide the evidence score based on all this informations. So you have another, like, you can search the biomarker, cancer type, level of predictive implications of therapy from the molecular oncology almanac. So that's funded by the National Cancer Institute and the Swedish Cancer Society, System Biology Cancer Genomics Cloud. The research on 70,000 cases is curated primary literature. So that's basically from the literature search. And you can see the recurrent chromosomal aberration using these databases. So this is all about this lecture. I know. So thank you very much. So if you have any questions. Mohsin Manan, that's a good question. I'll get back to you later on. There are some data.
If I'm not wrong, I think there's a database known as
Yeah.
Yeah.
I found one, yeah. So yeah, I will upload the lecture of, I will upload the slides after this lecture. And, yeah. No. So could it be inferred that the variation is pathogenic? If it's no, not necessarily. So it has first, the first evidence, it should come like it should be experimentally validated. If not, you need to look at the whether that has any like implications of the protein structure, kind of mutations.
So thank you, Maksud. Thank you very much for wrapping such a vast genomic area topic in just an hour. So I know it's really very difficult, but for all the participants, I think it's going to be very beneficial for their projects or in order to understand the basics of population genomic databases. So might be there are so many questions ringing in your mind or might be if all the participants watch the video. So if any question, query, comments, suggestions, recommendations came up, all the participants can write into the Google Classroom. They can discuss, ask at their own ease. So we will be there to answer all your queries and comments. So I have an announcement to make. As far as our schedule is concerned, so we are completed with our schedule because on the 12th of October, we have this either discussion day or the Maksus lecture. So so last week we already have had a discussion day, and this week we have a Maksus lecture. So for the next week, 19th October, because that week is going to be the last week of our Essentials of Bioinformatics, our virtual course. Next Tuesday, we have a national holiday in Pakistan. So we are trying to put up a concluding session either on 18th October, Monday, or if it's not possible, might be the end of the week starting from the 18th of October, or might be the next Tuesday after the 19th October. So we are not sure right now, but I will announce at the Google Classroom, and I will drop an email to all the participants, because the concluding session will be hosted by one of our MOMI collaborators from the Stanford University. So it's about the basics of multi-omics data. So it's going to be a very basic lecture based on the cutting edge scientific technology which we are used nowadays, how to use all the omics data at one platform. But the date is not announced, but I will keep you posted. Maksud, I'm not sure that Dr. Baki is available on 18th October or not, but if you drop an email to Sayyed, or so that he can book the slot, because I really want that all the organizing committee members have to say some words before the start of our concluding lecture, and then we have a lecture of 50 to 45 minutes, and then we will wrap up with a happy note. The date is not announced yet, but I will keep you posted all. Okay.
Oh, good, yeah. Thank you, Wakasim. I'll give them. I'll inform them.
Definitely. So if there's any question you can ask within a minute or so, otherwise we will log out. Okay, so thanks to all the participants. I will update you. Thanks, Akbar. You can stop the recording. Thank you all.
Thank
See
you.
you.
Thanks, everybody.
Bye bye.